Hello everybody and welcome to our first video. So in this video we are going to be going over biochemistry chapter 1 for the MCAT. And so let's get started. So chapter 1 of biochemistry really focuses on amino acids, peptides, and proteins. So one of the first things we're going to talk about are proteins. So proteins are made of monomer amino acids. So a peptide is actually a chain of amino acids. Now, let me get my other color here. So if we have our amino acid right here, this is just our, general structure of an amino acid. So we have our alpha carbon, we have our hydrogen, we have our carboxylic acid, amine group, and we have our R group. And we're going to talk about the R groups in a later part of the video. But those four parts, okay, all four of those make up the general structure of the amino acid. And so this is a monomer. This is just one amino acid. What happens is you get a protein from peptides. So peptides, as we said, is a chain of amino acids. So what happens is through a condensation or a dehydration reaction, you're going to get this. It's going to start growing a peptide chain. So condensation or dehydration reaction. And right here, okay, you can see this, and here's the second amino acid, but they're joined by a peptide bond. So this peptide bond actually produces water. So what happens is you take this OH from this amino acid, and let's say you have another amino acid over here, okay, and etc. The R group, and then you have your COO, OH, okay? So what's gonna happen is you're gonna take your OH from there, and you're gonna take your one of your H's from here, and this is gonna produce water, which is why it's called a dehydration, because you're taking this water molecule out. And that is what forms this peptide bond right here that is connecting these amino acids. So the amino acids are synthesized. This peptide chain is synthesized from N to C terminus because you're adding to the carbon end. That's how you're adding to your peptide chain. So another thing to note is there are 20 amino acids that you need to know for the MCAT. And we will, as we said, we'll get into the side chain shortly. So all of those amino acids, except for glycine, are chiral. And chiral means that they have this alpha carbon that is connected to 4 different substituents. And we will see that glycine is the only amino acid that we're studying that does not have that. So all of our amino acids except for glycine are chiral. And we have all of our amino acids except for cysteine are in S configuration. And we will go into configuration in a later chapter as well, but that's just important to note that all of the amino acids are S configuration except for cysteine. And then finally, what we're going to talk about in this portion of the video are these side chains. So next we're going to go into our structures of all of our 20 amino acids, and we're going to be grouping them into polar, nonpolar, aromatic, acidic, or basic side chains. And when we say side chains, we're talking about this R group right here. So this R group is what gives the amino acid certain properties that will make it fit into some of these categories. All right, so let's keep going. So hello everybody, and let's continue on with our amino acids. So first we're going to start with our nonpolar amino acids. And there are 7 of those, and it is very important to know all of the structures of the amino acids for the MCAT. Their full names, their structures, their properties, their first letter abbreviations, and their 3-letter abbreviations. Okay, so let's get started. So the first thing we have is, the first amino acid we have is glycine. And I'm going to be drawing the side chains in blue. So all of the amino acids will have this carboxyl group, the amine group, and there's also a hydrogen here, and an R group. And so that's what we saw in the first part of the video. So I'm going to be drawing the R groups right now, and it's the R groups that make these amino acids nonpolar. So the first one is glycine. And so for glycine, it actually has 2 hydrogens. So one of the hydrogens is there as a regular, part of the amino acid structure, But then glycine actually has another hydrogen added as its R group, which is, as we mentioned before, is the only non-chiral amino acid, because this alpha carbon is not connected to 4 different substituents because 2 of the substituents are the same. So our next amino acid is valine. And so valine, valine is a little easier to remember because it kind of looks like a V. Okay, so this is valine, V and VAL as its 3-letter. Sorry, and glycine is G and GLY. So this is valine. And the next amino acid, a nonpolar amino acid, is leucine. So we're going to come right up here. I believe you can see it. Let me move it over a little bit. There we go. So now let's look at leucine. So leucine actually has, I'm going to write it over here and then I'll move out of the way. So leucine kind of looks like this. It has 1, 2, 3, 4 carbons in its side chain attached to that alpha carbon of the amino acid. Next we have isoleucine, and isoleucine has I and ILE as its 3-letter abbreviation, and its structure is like that. That is isoleucine's R group right here. Okay, so our next one is proline. I might have to move the camera a little bit for these ones, so just bear with me. Okay, proline, I'm gonna draw it and then I'll move it. So proline is also a very interesting one. So proline, down here, which we will see, actually has a ring-like structure attached to it. So you will know that this one is proline. I will draw it and I will move out of the way. Okay, so proline looks like that, and it has P and PRO as its 3-letter abbreviation. Okay. Next we have alanine. Alanine is, definitely one of the easier ones to remember. Just move this back here so you can see. Okay, so there we have alanine. Alanine just has a line to represent the one carbon attached to it. And finally, we have methionine down there at the bottom. It's a little more complicated. So methionine actually has a sulfur in this R group. So this is what methionine looks like. And I like to remember methionine because it kind of looks like an M with the sulfur in it. So those are our nonpolar amino acids. And stay tuned for the next group that we're going to go through. Hello everybody, welcome back. So now we're going to go through our polar amino acids, okay? So our polar amino acids, again, their side chains, what we're about to draw out here, is what makes them polar. So the first polar amino acid is serine. So serine, I'm going to draw the side chains in blue. So serine, S-E-R-S, actually just has an OH group as its side chain, okay? And that's what makes it polar, is this OH group. So next we have threonine, and threonine has an OH group, there we go, with an extra carbon over here. So that is threonine, THR and T as its 1-letter abbreviation. The next one we have is glutamine. So glutamine is a 1-letter abbreviation, it's a little weird, it's Q, and that's kind of one of the odd ones that you just need to remember. So glutamine is Q, and its side chain, H2N, so it has another amino group there, and another carbonyl carbon. Okay, so that is what glutamine looks like. And then we have asparagine, which is very similar, but just with one less carbon. So the asparagine is going to look like this. Carbonyl carbon. Drew it a little bit further away there. Sorry. But yes, it just has 1, 2 carbons. And here we have 1, 2, 3 carbons, okay, in that chain. So, oh, and our last one down here, our last polar non-aromatic amino acid is cysteine. And cysteine is also a special one. It has HS with carbon. So cysteine, as we've mentioned, is also important to note. So that's an HS attached to a carbon, attached to the carbonyl, attached to the alpha carbon. So please stay tuned for our next group of amino acids. All right, so for our next group of amino acids, we have our aromatic amino acids, okay? So the first one that we're going to talk about is tryptophan. Tryptophan is the largest, is the amino acid with the largest side chain. So this one is a little more complicated. So just give me 1 second here to draw it out. So you're going to have, that's a nitrogen. All right, so this right here is tryptophan, and it's aromatic due to its aromatic ring right here, okay? So tryptophan is TRP and W, another one of those weird ones, but my friend did make up a really cool acronym for this. So if you go on a, let's say you win a trip, okay? So that's how I remember this one, trip, you would win a trip to go on vacation. So all the vacations you want after we're done studying for this test, right? So phenylalanine, is our next aromatic amino acid. This one is quite simple in comparison. And so we just have an aromatic ring right here. Just like this. All right. So that is phenylalanine. PHE is its 3-letter abbreviation, and F if it's 1 letter, but phenylalanine, for F, yes, that makes sense as well. And then, finally, we have tyrosine, and tyrosine has an aromatic ring with an OH group attached. So when we see the OH group, if you remember the last group that we did, the OH group meant that that group was polar, right? So tyrosine is the only polar aromatic amino acid. And I do have a song to remember all these amino acids, and maybe if I have enough courage to do so, I will post the song. But you know, it's really important to know the structures as well. So tryptophan, phenylalanine, tyrosine are the three aromatic amino acids that you need to know for the MCAT. Okay. All right, everybody. Now here is our last group of amino acids. So we are now working through our charged amino acids. So you'll have positively or negatively charged amino acids due to their side chains being acidic or basic. So we're going to go through exactly what that means in the next part of the video. But right now, this is just the structures of the basic and the acidic amino acids. So the first basic amino acid that we're going to look at is lysine. So lysine has LYS as its 3-letter abbreviation and K as its 1 letter. And the structure of lysine looks like this. Okay, so there's a few, carbons and then this nitrogen group right there. All right, so our next basic amino acid is arginine. Arginine has ARG as its 3-letter abbreviation and R as its 1 letter, and that's kind of easy to remember, arginine, and it has an R as its letter. So arginine side chain is like this. Oh, I don't know if I have enough space there. Let me just start over. So arginine side chain has a nitrogen there, a nitrogen there, as well as a nitrogen there. So I'm hoping you can see. Let me just move in here for a second. So that is arginine right there. It has a few carbons, a nitrogen, hydrogen, another double bond to a nitrogen, and then finally another nitrogen with a hydrogen. It's with 2 hydrogens, sorry. So yes, that was what our arginine was looking like. And then we have histidine. So histidine is actually a pretty cool one. So histidine is it has HIS as its 3-letter abbreviation, H as its 1-letter abbreviation, and its side chain kind of looks like a house, and that's how I remember histidine. So it has a nitrogen down here, nitrogen up there. Okay, this is double bonded as well. But this kind of to me looks like a house. So that's how I remember histidine. I'm not sure if you can see it. There we go. All right, so this is what histidine looks like. Okay, so all of our basic amino acids have a positive charge. Okay, and like we said, we're going to see how they get those positive charges at a neutral pH in the next part of the video. But for now, those are your basic amino acids. Now we're going to get into our acidic amino acids. So our acidic amino acids will have a negative charge, and those there are only 2. So one is aspartic acid and one is glutamic acid. And again, it's right down here at the bottom. So I'm going to fill them out and show it to you. So feel free to speed up the video at this moment. All right, so let's take a look at glutamic and aspartic acid. So aspartic acid looks like this. Okay, it has carbon, carbonyl, oxygen, OH, so it has its carboxylic acid side chain over here. And then we also have that for glutamic acid, but glutamic acid just has 1 extra carbon in the chain. So these both are our acidic amino acids, and those will have a negative charge. Another important thing is aspartic acid has ASP as its 3-letter abbreviation and D as its 1 letter. So aspartic acid is how I remember the hard D's in there. And then the glutamic acid, and it has an E. So that's a little bit hard to remember, but you can just remember A, B, C, D, E. Okay, so that's how I remember those. All right, now let's keep going here, everybody. So as we continue through chapter 1, we have our peptide formation, which again is through condensation or dehydration reaction, because you are removing a water molecule and as you do this reaction to form your peptide. But to break a peptide, this would take a hydrolysis reaction, and you're using water, hydro, to break the bond, so hydrolysis. All right, so we also have different structures for our proteins. One being the primary structure is the linear sequence of amino acids. And again, this is held together by peptide bonds. Now we have our secondary structures, which are the local structures of neighboring amino acids. And so this is typically our alpha double helix or our beta sheets. And this level of protein structure is held together by hydrogen bonds. And one important thing to note here is proline actually would not likely be found in the alpha double helix but proline would likely be found in a beta sheet due to its structure. It can help with the zigzag nature of the beta plated sheets. So our tertiary structure is down here. So that is the 3D shape of the polypeptide. So it interacting with itself. And this can happen through disulfide bonds, hydrophobic interactions, acid-base interactions, or hydrogen bonding. So that all can play a role in tertiary structure. And then we have our quaternary structure, which does not always occur. Now, the quaternary structure is the interaction between the peptides in different subunits of a protein. So some proteins maybe are made of different subunits, and that is where the quaternary structure would come into play. Another thing to note is denaturation. So some specific types of detergents or heat, different levels of pHs, those types of things can denature the protein structures, the levels of proteins. So there will definitely be some questions about that as well. Another important thing to note is a conjugated protein. So these are proteins that get their functions based on the prosthetic group covalently attached to them. So for example, lipoproteins are proteins that have a lipid prosthetic group attached to it. Glycoproteins are proteins that have a carbohydrate covalently linked to it. And a nucleoprotein is a protein that has a nucleic acid prosthetic group. So there's just also important things to note. And a good example that they do use in one of the MCAT books, it talks about heme. So each of hemoglobin subunits, so again, that would play into our quaternary structure because heme has different subunits, contains a prosthetic group called heme. Okay, so that heme group actually contains the iron that binds and carries the oxygen, in our hemoglobin. So, but the hemoglobin is actually inactive without the heme group. So that is why these prosthetic groups are so important. So that is all for our protein types. And next we are going to our protein levels of protein structure. And next we are going to talk about why some amino acids are charged, or why they are given, why they belong to the acidic or basic amino acid group. So that's up next. All right, so as we're looking at our example of a basic amino acid, we're looking at lysine. So pictured right here is what our lysine would look like at a pH of 7. As we know, the basic amino acids have a charge of +1 at this pH because we have a plus charge here, negative charge here. So technically those two charges would cancel out. We are left with a positive charge due to its R group. So an important thing to note once again is the 3 pKs that exist for this molecule due to its 3 charged side groups. So if we are moving this molecule into a more acidic solution, as we've previously seen, this solution would give an H to this COO minus group, and it would become COOH, right? That means that lysine would have a charge of +2 in an acidic solution because this charge would no longer exist, and we have a positive here and a positive charge there. So lysine, as we said, would have a charge of +2 in an acidic solution. As we move on and continue into a more basic solution, so we would, this would again become deprotonated as we're increasing the pH. And then we would be in a more basic solution. So this group right here would, as we saw, lose this positive charge because the hydrogen would be donated to the solution to create water. And then we're going to be left with this negative charge of the deprotonated COO minus group and this positive charge of the NH3 plus due to its R group. And so this is where we would find our neutral a molecule for lysine, it would be in a more basic solution, and that would make sense as we showed for the other example. If you're taking the pKa of the amine plus the pKa of the side chain divided by 2, that makes sense because both of these, pKa of the side chain and the pKa of the amine, would be in the more basic range, which makes sense for the pKa for those molecules to be higher, right, in between the two. So now if we take it one step further, so this would be when lysine is neutral and has no charge. But if we take it one step further, we see that again, this side chain would then become deprotonated, and we would be left with this -1 charge. So that is where lysine would have a negative charge, would be in a very basic solution. So hopefully drawing these out really helped you understand. I know it really helped me go through things when I really drew them out and saw all the different possibilities that the ways this molecule could look through the different pHs. You could also look for different titration curves. This is, kind of under those categories. But really understanding what happens to the molecule at those different pHs is really going to help you conceptualize things. So thanks for watching, and stay tuned for the next video.